Now that we have gone over the basics of protein structures and some of the motivations for studying them, let us take a look at the technological advances that allowed for us to visualize these structures in such great detail. In this section of the lecture, we will look at how protein structures are determined through one of three techniques and discuss how protein shape is predicted when they have not been experimentally determined. First, let's take a brief look at the history of structural biology. In the early 20th century, the first crystallographic studies were conducted on salt crystals. 43 years later, in 1953, X-ray crystallography was famously used to determine the double helix shape of DNA. 5 years later, myoglobin became the first resolved protein structure. As X-ray crystallographic techniques were refined, larger and larger protein structures could be resolved, culminating with the structure of the ribosome being solved in 1999. In parallel, another technique, cryogenic electron microscopy, or cryo-EM, which has become revolutionary in the field of structural biology, began to develop. This technique was first applied in 1975 to image bacteriorhodopsin. Improvements throughout the 80s and 90s by three scientists gained them the Nobel Prize for Chemistry for their development of cryo-EM in 2017. Continued improvements in cryo-EM throughout the 2000s has allowed us to achieve very high resolution images of very large proteins and ones that are notoriously difficult to study, such as membrane proteins. Throughout the 1970s, it became feasible to visualize these structures using computers. This led to a wide development of computer modeling and simulations in molecular dynamics. To gather all these protein structures, the protein data bank known as the PDB was created in 1971. Hosting all the available protein structural information, the PDB effort is vital to the structural genomics efforts of the 1990s and present day, which is to resolve all structures for all proteins. Today, the PDB hosts over 161,000 macromolecular structures and is fastly becoming a one-stop shop for all data related to protein structural properties. I cannot stress this enough. The PDB is every structural biologist's homepage. Let's quickly look at the workflow required to resolve a protein structure. First, the sample must be prepared, and this usually involves protein expression, purification, and some additional preparation, depending on what method is going to be used for the structure determination. These methods are X-ray crystallography, nuclear magnetic resonance, or electron microscopy, which we'll discuss in detail on the following pages. Finally, once the structure is determined, it is deposited in the PDB and can be used for understanding biological mechanisms. The first step in determining protein structure is preparing the sample. This requires that the protein of interest is expressed in large amounts. Then, the protein must be purified and isolated from other proteins. This usually involves a lot of centrifugation and affinity chromatography. Once purified, additional preparation is necessary, depending on which technology will be used to resolve the structure. For example, X-ray crystallography requires that the protein be crystallized in its native state. Nuclear magnetic resonance requires that the protein is solvated in an aqueous solution. Cryogenic electron microscopy, or cryo-EM, requires that the protein is vitrified or quickly supercooled. The most common method of determining structure is X-ray crystallography. Below are two resolved protein structures. Can you predict which one is difficult to prepare? If you guessed the protein on the left, then you are correct. As you can see, this protein has hydrophobic and hydrophilic domains, and for this reason, crystallization of this protein in its native form in aqueous solvent is extremely difficult, if not impossible. In the following slides, we will go through each of the three methods of resolving protein structures. The most common and oldest method of resolving protein structures is that of X-ray crystallography. Following the long and often difficult process of crystallizing the protein in its native state, the sample crystal is isolated and subject to a beam of X-rays. As the rays hit the sample, they are diffracted onto a detector which measures this pattern. This diffraction pattern yields an electron density map, with high density regions where the beam was most diffracted. Using this density map, the atomic model of the protein polypeptide chain can be refined and fitted until the structure is resolved. To appreciate how far X-ray crystallography has come since its inception, let's take a second to appreciate its significance to the field of structural biology. Most notably, the first contribution came when the double helical structure of DNA was determined using this method by Watson, Crick, and Franklin in 1953. This achievement would earn them the Nobel Prize in Physiology and Medicine. In 1960, Perutz and Kenji resolved the first protein structure, myoglobin. This achievement would once again earn its investigators a Nobel Prize. Following improvements in resolution, sample preparation, and beam technology, the large structure of the ribosome was finally resolved in 1998 at a resolution of 9 angstroms. This structure would subsequently be improved to a resolution of 2 angstroms. This achievement once again earned its investigators a Nobel Prize in chemistry. The second oldest technique to resolve biological structures is nuclear magnetic resonance, or NMR. Developed in the 1940s, this technique relies on the principle of nuclei spin. When a highly purified protein solution is subject to a magnetic field, the magnetic moments of the nuclei can align with the field. Using pulses of radio waves, nuclei will resonate at their characteristic frequencies, identifying the amino acid side chains. When expanded in multiple directions, the distance between these characteristic atoms can be measured and uses constraints to fit an atomic model. Because of the constraints on the residues, different viable models are often superimposed on each other to resolve the structure, as seen here to the left. The newest technique to resolve structures is called Cryo-EM. Here, an electron beam is fired at a protein sample. The scattered beam is then magnified through a lens to create an image on the detector. From here, the structure can be determined. Initially, the technique to use electron microscopy for biological molecules had its limitations. One of them was that the energy of the electron beam often irreversibly destroyed the protein sample. Prior to the vitrification technique developed by Dubochet, samples would often dry out during experiments and denature. Lastly, the imaging software developed by Frank, in which blurry traces of a protein are first separated from the background noise, then reoriented and grouped together by shape to form 2D images, and lastly, critically assessing their differences to generate high-resolution 3D models, was absolutely crucial to the impact this technology continues to have in structural biology. The most important thing to realize is that cryo-EM does not require the sample to be crystallized in solution. Rather, the sample is vitrified in its native state, allowing for extremely large proteins as well as membrane proteins to be resolved. This will, yeah, that's fine. As we did with X-ray crystallography, let us briefly marvel at the progression of cryo-EM throughout the years. In 1968, de Rosier and Klug were the first to image a bio-macromolecule using an electron microscope. Building off this work, Henderson was the first to use electron microscopy to create a 3D model of a protein. In the 80s, Frank's image analysis innovations drastically improved the resolution of this technology. And lastly, the vitrification technique developed in 1984 allowed for cryo-EM to bypass the bottleneck of crystallizing large or even membranous proteins. Using these improvements, the 1975 model was refined. Not bad, eh? Lastly, let's take a look at the capabilities of cryo-EM today. Pictured on the right is a bacterial efflux pump containing over 12 protein subunits and spanning both cellular membranes. The most important aspect of any protein structure is the resolution at which it can be determined. This is analogous to the quality of the structure. As you can see from the video, increasing in the resolution value creates larger pockets in the electron density map, making it more and more difficult to determine exactly where each specific amino acid residue is present at any given 3D coordinate position. On the right, we can observe the same effect. What happens when we don't have an experimental structure for an amino acid sequence we are interested in? In these cases, we become systems biologists that are satisfied simply with the predictions of what the structure will look like. To predict a protein structure from sequence, we can use two methods. The first is template-based modeling. Here, we take a sequence and map it onto another structure in the PDB. If the sequences overlap sufficiently, we can fit our sequence into the density map of the existing structure. The second method is modeling protein folding subject to energetic constraints. This method is much more computationally intensive than the former. Luckily, it is often the case that protein shape is often more conserved than genetic sequence. Taking advantage of this and the sheer number of experimental protein structures available, template-based homology modeling methods often result in fairly good predictions of protein structure. The last step involved in determining protein structure is its deposition into the PDB. As we mentioned earlier, the PDB is the website that hosts almost all protein structural data. Not only a database of depositions, PDB connects and interfaces with a variety of other databases and information. Pause the video here and go explore to learn more about the different structural databases.